Good morning, my name is Samantha Riles, Communications Officer from Sugar Research Australia, and I'd like to welcome you to today's webinar. Please note that today's meeting is being recorded and will be available on our website. At any time that you are not speaking, can I please ask you to remain on mute to keep the background noise to a minimum? If you are having any technology issues, please send a message in the chat box, which is located on the menu bar of your screen, and I will be able to assist you as I'll be staying in as a co-host to ensure all runs smoothly. Thank you to those who have sent in your Q&A via email. At the end of the presentation, our researchers will be taking questions and we encourage you to take an active part. You can type questions into the chat box throughout the presentation and Claire will moderate these at the end. Alternatively, if you have questions, please unmute your microphone and ask our researchers. I'd now like to hand over to SRA's Executive Manager of Communication, Brad Pfeffer, to discuss the format for today's webinar.
Thanks, Sam. So this webinar is a bit of a follow-up to the one that Jared presented about a month ago about yellow canopy syndrome. And the objective of this webinar is to talk a lot more broadly about some of the various factors that can cause yellowing in sugarcane, because after that webinar, we had a number of questions that came to us from right across the industry. So we thought it was a good opportunity to bring people together to talk about some of those questions and go through them all. So just to kick off things today, Jared Scalia is going to give a bit of a recap on that previous webinar, just in relation to identification of yellow canopy syndrome. And then we're going to switch gears and go over to Claire, who's going to moderate a few questions. And then we'll be able to hear from people in the room as well if you've got further questions. So I'll pass over to Jared, who will kick things off with that recap.
Thanks very much, Brad, and thank you everyone who is attending this morning for this Q&A webinar. So yes, as Brad said, I just want to recap on the symptoms of YCS that were outlined in the previous webinar. So what is YCS? So YCS is a condition describing yellowing in the mid-canopy of sugarcane, and that's what makes it quite different. You don't see this yellowing in the very young leaves above, and you don't see this type of yellowing in the older leaves below the mid-canopy, and I'll come to that in a little while. When do we see YCS? Well, it occurs during the peak growing period, and that growing period is between December and March, and it's also the time when it's the highest rate of photosynthesis and light intensity. So during this presentation, whenever you hear me say the word photosynthesis, just think of it like being a sugar factory, the factory that's producing the sugar or sucrose. So that's the time of the year when the majority of the sucrose is being produced, and it's also the time when there's the highest light intensity and the highest growth rate, and that's a very important thing to remember. Next slide, thanks, Brad. Okay. So how is YCS identified? Well, we need to have all of the following conditions met for you to be confident it's YCS. So first of all, it has to be in the mid-canopy. Yellowing has to be in that mid-canopy. The symptoms have to have a particular pattern of development, and they also have to have a golden yellow colour. And I'll talk more about that in a second. And the leaves have to also be very high in sucrose content. So their sugar content is always high. This without question. YCS leaves always have an abundance of sucrose. Because they have an abundance of sucrose, the plant tries to offset that by producing another product called starch. And that starch is stored in the leaf, in different parts of the leaf. But during the night period, starch and sugar get used for energy when the sun isn't shining, when that photosynthesis isn't occurring. And by first light in the morning, both sugar and starch should be at around about 0 levels in a healthy plant. However, in YCS plants affected by YCS, in the morning, that starch level is still quite high, and so too is the sucrose level. But we can do a simple stain test for that, and that's what we have developed, and it's out there being used in industry as I speak. So that's a good way to get a visual. So let's have a look at, in the next slide, some of the symptoms that you would see on the leaves. Okay, so I said before it has to occur in the mid-canopy. When we look at our crop, we can divide it into 3 zones, an upper, mid and lower. The upper zone is the area of the canopy that's very immature and it's still actively growing and developing and maturing. And the mid canopy is the area where we see YCS, and the lower canopy is the aged area, the older leaves that have pretty much done their work and they're spent. So they're starting to senesce or die. The mid canopy is the powerhouse or the work zone really of your crop, because this is where they produce the majority of the photosynthate or produce the majority of the sucrose. These leaves in here are your workhorses. So like leaf 3, 4, 5 and 6. Leaf 4 and 5, leaf 3 and 4 is where we typically see the majority of YCS symptoms when they first start. But we have seen it in 3, 4, 5 and 6. So we must get the zone correct to identify it. Now, I just want to point out one very important thing while we're on this slide. To identify YCS, to be sure that you've got YCS, it's imperative that you pick it up very, very early at this stage. When you look at your crop and it's all green, that's fantastic, but you've got to keep looking. If you see the start of yellowing in the mid-canopy region, in that leaf 3, 4, 5 and 6, that's the time to keep an eye on it and follow its progress. When it starts, you'll see a little bit of yellowing in one of those leaves, and there will be green leaves beneath it and green leaves above it. That's because those leaves aren't old. They're not senescing, they're not starting to age and die naturally. And yellow canopy syndrome will work its way up the canopy, not down. So if you come in and pick it up late in its maturation or in its symptom expression, what will happen if you think about it, if it starts in a leaf 4 and leaf 4 becomes more and more yellow and then it moves up to the leaf above it to leaf 3, eventually that leaf 4 becomes a leaf 5 and then it becomes a leaf 6. And as it ages, it'll join up with those older leaves below. Then you'll have absolutely no way of knowing whether you've got YCS if you pick it up late, because it's all joined up. It makes it really difficult to identify. So the point I'm trying to make here is you have to pick it up early. For those of you out there who are involved in sugar services, you have to pick it up early in the piece. You get there too late, it's very, very difficult to identify. And because of high sucrose in the leaf, which I'll talk about shortly, a lot of secondary effects come in over the top. Okay, next slide. Thanks, Brad. All right, so at the top of this slide, we have a healthy, unaffected or asymptomatic green leaf, which is what we love to see. When YCS starts, in the second image there from the top, we see a little bit of yellowing occurring on one side of that white midrib, the center vein. And yellowing doesn't normally start towards the tip or the base. It normally starts around about the middle of the leaf, and it starts close to the midrib, and it becomes more and more yellow, and it moves, that yellowing moves towards the tip and towards the base and out towards the outer margin of the leaf. And eventually what we see a lot of, and I'm sure you've seen it as well, is that you get yellowing down one side of the leaf, on one side of the midrib, as you can see in the third image. That's very, very typical. And then finally, when it becomes really advanced, you see it, you possibly may see it advance onto both sides of the midrib. So why does it start sort of around the middle, and why does it move in the way that it does? Well, I said before that YCS occurs during the peak growing period. That's when it's producing the most sugar. It's also when there's the highest light intensity. So yellowing can occur anywhere on that leaf if it has high sugar and high light intensity. So if the leaf is a typical leaf and it bends in the middle, that's where it receives most of the light. And that's where it'll be producing the most sugar, and that's where you will see yellowing commence. If you've got a variety that's got leaves that stand more upright and the tips of the leaf are more exposed to light than further down where it's more shaded, then you typically will probably see yellowing start more towards the tip and so forth. So that's a very important point as well. You need more than just sugar. Okay, so why does the yellowing occur? I've talked about this is a time when we produce most sugar and that YCS leaves have high amounts of sugar in their leaves. YCS affected plants have high amounts of sugar in their leaves. Well, it's not the sugar per se that actually causes the yellowing. It's the thing that triggers it. Now, you might think that all this problem with the yellowing starts in the leaf because that's what you see. But we found out through our research that it actually doesn't. There's nothing wrong with the leaf. The leaf is healthy. It's not healthy when it goes yellow, that's sure. That's true. Well, so why does it go yellow? And why does this sugar accumulate? And that was a very big part of the research. Why wasn't the sugar leaving the leaf at a really good rate? And last time in the webinar I spoke about needing a strong sugar gradient between the leaf and the stalk. The first thing that came to mind was that there must be a blockage, a big blockage in the pipework carrying the sucrose or the sugar from the leaf to the stalk, particularly to the two internodes beneath that leaf. So we looked for that. We couldn't find any consistent evidence of a major physical blockage caused by a pathogen or any other plant response to a particular stress that could slow down the sucrose enough to cause the buildup. And I talked about gradient last time along the leaf, and we saw a transition where we didn't see a buildup in one particular part of the leaf more towards the tip than we did it towards the base. And so that we also found that sucrose was being loaded into the pipeworks. It wasn't stuck at that point, and it was transported, and it ended up in the stalk, and it ended up in those internodes beneath the symptomatic yellow leaf. So what we found was that there wasn't the cause of the sugar buildup in the leaf wasn't caused by a problem with the leaf. It was caused by a problem with the stalk. And that problem was that the warehouse, so to speak, or the receiving part of the stalk, the two internodes that receive 80% of the sucrose from that leaf, were just too small or had a sluggish metabolic rate. They couldn't accommodate all the sucrose being produced by the leaf. Think of it like this. You own a pizza shop, COVID-19 comes along, there's no orders for pizzas, so you don't want to keep producing more pizzas. But the delivery guy keeps bringing you the product to make the pizzas, and they build up in your factory, in your restaurant. It's a little bit like this. When sugar builds up in the leaf, it's pretty smart, and it's got sugar sensing molecules in there that tell the genes that control photosynthesis to turn down or to turn off, not to produce any more sugar or sucrose because there's too much there. And that's a good thing to do. You don't want to keep making more sucrose because the problem will get worse. Unfortunately, there's one flaw to that system, that when the factory is turned down, in this case, the product that comes in to build those pizzas, help build the pizzas, in the case of sugar, it needs water and it needs sunlight and carbon dioxide. You can't turn off the sun. The leaf has no way of turning off the sun. Sun keeps shining, and that energy hits the leaf It's transported through the machinery, the photosynthetic machinery inside the factory, and the leaf can't use it because it's turned down the genes that control the factory. That energy has to go somewhere, and unfortunately that energy burns the leaf up from the inside, and that destroys the green pigment in there, and that's what causes the yellowing and the other pigments to come through, and they have a really golden yellow colour, and those other pigments make the colour of YCS unique. So that's why we see the yellowing that we do, and that's why we see it where it is, and that's why sugar builds up. It also tries to do other things, of course, to counter that by putting the carbon into other places, like starch. Okay, so can we go to the next slide? Thanks, Brad.
No, it was heavily intoxicated, probably even partially concussed. He had no idea what was going on.
I'm sorry, I missed the first part of your question. I apologise.
Is that just the sort of bloke he is? You're absolutely right. The sort of person he is, irrespective of whether it's somebody on peaceful protest side or somebody, in this case, on the antagonistic side, I would say. We were there and Patrick was.
I really apologise. There's so much echo in your question. I really haven't been able to pick it up. I wonder if you could type it and send it through to Sam, unless someone else in the group can relay it to me.
Yeah, I think that's a good suggestion. I couldn't hear it either, unfortunately. So yeah, if you'd like to type your question, we'll get to it when we get to questions.
Apologies for that. Okay, so the next slide that Brad's put up here is, if we move along with the webinar, is about what are some of the other causes of yellowing that we see in canopy of sugarcane. So in this slide here, and as you are all aware, other things can cause yellowing like drought and herbicide damage, insect damage, disease, nutrition deficit, and just natural aging or senescence. And it's important to understand how these symptoms that we commonly see differ to YCS. So here we have 6 images, labelled 1 on the left through to 6 on the right, and it's got leaves with different amounts of yellow, or different types of yellow, I should say. And if I asked you to identify which of those was YCS, I think you probably would have some difficulty, because I'm having difficulty identifying YCS in there. But one of those images was taken from a YCS field, YCS symptomatic field. So make your own guess, and I'll give you the answer shortly. But remembering the things that I said before about the colour and how it sort of starts on one side of the midrib and those sorts of things. Well, we'll go through them. The first one actually is damage caused by whitefly. The second one is a condition called yellow leaf syndrome. The third one is aphid damage. The fourth one is actually YCS. Fifth one is sugarcane yellow leaf virus. And the sixth one is water stress. So the point being that it's very difficult to identify YCS, as I said before, with all the other yellows going on out there, and it becomes very confusing. And there are many more yellows as this, which we're going to discuss shortly. But if we can pick up yellowing early, we've got a better chance. If it's in the mid-canopy and all of those criteria that I discussed, you've got a better chance of identifying YCS, and there's less chance of misdiagnosis. Thanks, Brad.
Thanks, Jared. Barry's now going to talk to a couple of slides about some of those other yellowing, just very briefly, before we get into questions.
Okay, thanks, Jared and Brad. Yeah, I thought we would talk about nutrient deficiency and some of those symptoms, just briefly, I guess, because obviously we could spend probably this whole meeting just talking about that issue. So I think the key points are that nutrient deficiencies can cause yellow symptoms, but those symptoms are different to what we typically see with YCS. And so the best information I can actually provide is in the Australian Sugarcane Nutrition Manual, which you can find on the SRA website if you go into the nutrient management section. And it covers off on all the nutrients. It has pictures like you can see here of some of the pictures of the symptoms. But there's also some good tables in there that compare the symptoms from all the different nutrients. Briefly, if we look at these two here, which is nitrogen deficiency and potassium deficiency, I guess with nitrogen, what you tend to see is just a slightly paler color, paler green or slightly yellow. It does start in the older leaves as well, so it's similar to YCS in that regard, but you just never see that sort of bright yellowy orange color with a nitrogen deficiency. So we have trials where we've applied no nitrogen for 6 years, and we see a slightly paler green color, but not that sort of bright yellow that you'll see with YCS. Potassium deficiency, initially when YCS first started, we did consider potassium deficiency. The symptoms in terms of the color are fairly similar. They do start in the older leaves first as well, but the key difference with potassium deficiency is that it will start at the leaf tips and the leaf margins and develop from there. So you'll see it refers to it as a burnt type appearance. So it's different to YCS in that YCS, as Jared pointed out before, is tending to start in blotches, I guess, in the middle of the leaf and then spreads from there, whereas potassium deficiency is starting at the tips and margins and working from there. Next slide, Brad. Okay, and then we threw in some other ones here. So you can see iron deficiency, copper deficiency, and zinc deficiency. They all have different but distinctive symptoms. So you can see there's variations of stripes and also a mottling type appearance for copper. I guess the key point for all of these ones is that they tend to start in the young leaves first. So they're very different to YCS in that regard. These elements, deficiency in these elements, you'll see at the top of the canopy first, not in the middle of the canopy or lower canopy. So again, they're very different to YCS. And so I won't go through every nutrient, but I guess the key points are for nutrients is that they'll differ in terms of where they occur in the canopy. So some will occur in the bottom of the canopy first and others at the top. And the symptoms will be different to YCS as well. But again, going to the SRA website and looking at that nutrition manual is the best place to find that information. Just to add to that, I guess, over the years of the YCS projects, nutrition was looked at YCS sites. They looked at soil sampling and also sampling the crop to look for nutrient deficiencies. The only things that really stood out was that silicon was actually increased, which tends to be associated with stress responses. And the other thing was that magnesium was slightly lower in YCS cane, but it was still above the critical value. And then that was taken further in trials where magnesium was applied to the soil and magnesium was also applied to leaves in trials, but there was just no response to that magnesium. So those were the only nutrient issues that were found and they were investigated and nothing really came out of it. So we don't believe YCS is a nutrient-related issue. Just some of the other symptoms you obviously see out in crops. Water stress, I guess we're all familiar with. Again, you will see yellowing with water stress. It tends to, again, start at the leaf tips and leaf margins. And those tips and margins will obviously die and turn brown as well. I guess the key difference between YCS and water stress is that those water stress blocks take on that typical sort of spiky appearance and the leaves will roll, whereas in YCS the leaves don't don't actually roll. They remain fully expanded. So you know, quite a difference between water stress and YCS in that regard. And I guess there's an image there of grub damage as well, which is similar to water stress, because obviously if the grubs have affected your root system, you're not supplying water to the crop. So the symptoms can be quite similar, but will tend to be more patchy in terms of they'll only occur where the grubs actually are, as opposed to water stress, which would tend to be more general. Next one, Brad.
So Rob McGarry is going to just talk briefly to this one slide.
Yep. Thanks, Brad. I'd like to welcome everyone this morning. I'm just going to talk very briefly to 4 diseases, or 4, actually not all diseases, but 4 examples of either diseases or a condition that can affect the canopy at the time of year when we would see yellow canopy. So the first 2, the first one's orange rust and yellow spot. And these can occur through the industry, but yellow spot, particularly in the wet tropics, orange rust can be right through the industry. The things about those 2 conditions are that it doesn't cause that orangey yellow type colouring of yellow canopy. They're more a browning of the canopy, and so usually it's fairly easy to distinguish those ones. They produce lesions on the leaves. It's not a general colouring of the leaf. It's individual spots that together form a background leaf colour. So yellow spot and orange rust, 2 leaf diseases, generally easily separated from yellow canopy. The next disease is chlorotic streak. Chlorotic streak occurs right through the industry. It's caused by a protozoan organism. It does give that creamy yellow type symptom, but it occurs in streaks. So if you look at the canopy of the cane, you'll see that there's streaks running up and down the length of the leaf. It's not a general coloration like yellow canopy. So though the color might be similar, the actual streaking gives the main clue that it's not yellow canopy. Chlorotic streak does come and go. It doesn't affect necessarily the whole canopy either, in the same way that yellow canopy does. The last condition is yellow leaf, what we call yellow leaf syndrome, YLS, and it's probably the closest one to yellow canopy. It does tend to occur towards the end of the season as the crop matures, and the difference is that it typically starts on the midrib. So where yellow canopy starts in, say, 1/2 of the leaf blade, yellow leaf syndrome actually starts on the midrib, and you get the same sort of orangey yellow golden coloration. And it is interesting that the sugar content in the midrib is actually elevated, just like yellow canopy. The sugar content and the starch content is elevated, and probably that's the reason why the coloration is similar, as Jared explained. The main thing about yellow leaf syndrome is that it occurs towards the end of the growing season, typically around May, June, July, as the crop matures, and it's focused on the midrib. It can extend into the leaf blade, but you'll see it first in the midrib in the older leaves. So generally, once you can know those several characteristics about yellow canopy as yellow leaf syndrome, it can be separated quite easily from yellow canopy. So I think if you look carefully, you'll be able to distinguish all those conditions, and there are local adoption officers who can help you to distinguish those things. Thanks, Brad.
Thanks, Rob. Just before we hand over to Claire for questions, I just wanted to make sure we introduce some of the SRA staff with us today on the webinar. We've already heard from Barry, Jared and Rob, obviously, but we've also got Kevin Powell and Priya Joyce on the line. Kevin's up at Moringa and Priya's at Indooroopilly. So yeah, I'll hand over now to Claire, who's going to start with some of the questions that were submitted over email over the last week or so.
Yes, good morning, everyone. Thanks for joining us. I've received quite a few questions in the last week, which has been fantastic. Some of the received questions have been very specific to circumstances, and these will require more detail and they can be discussed. There will be some discussion today, but also the panel will be getting back to a few guys that have submitted questions. You can use the chat function, which is on the right-hand side, or if you scroll in the middle of your screen, the chat icon will come up, or you can also raise your hand. So there's a little hand function, and I'll try and raise those questions. We've already got a couple of those questions at the end of the other questions that we'll start with. So question 1, there were a number of questions relating to stress and specific questions around stress. So this is one from a grower. So if the conclusion is that YCS is caused by stress, is the advice in other areas such as reef regulations or from older style cane farmers reacting to persistent low prices to cut back on fertiliser and water a cause of stress? Why not up the fertiliser and water to reduce plant stress? So there'll be a few speakers on this question, and Rob will start first.
This is answering the question about upping the fertiliser. Is that right, Claire?
That's correct. That's correct. Yes.
That one's probably for me, Claire.
Oh, okay, I've got all three of you guys down there, but sorry, thanks, Barry.
No, that's all right. Look, like I said before, there's no evidence that this is or YCS is a nutritional issue, and so we really feel that sticking to your normal fertilizer plan or your nutrient management plan through the 6 easy steps system is the way to go. There's no evidence that increasing any particular nutrient will actually make any difference, because none of them have been shown to be deficient in YCS cane, and so we don't feel that you'd get a response to those, and so it just comes down to the economics, I guess, of applying extra nutrient for no benefit, and obviously there's the reef regulations to consider as well. So I think that the main point is to stick to the 6 easy steps guidelines, and that is sufficient to grow a really good productive crop.
Barry, while I've got you there talking, we have had a question from Andrew. The soil pH, is that also a clue for nutrients?
So yeah, I'm not sure if that's Andy Humphreys or not, possibly. So I guess one of the one of the interesting things about YCS is that we're seeing it in both acidic soils in regions like Mackay and the wet tropics and so on, but also in alkaline soils in the Burdekin. And so you'll tend to get different nutrients be more available on alkaline or acidic soils, so they won't be the same. So what that tends to suggest is that it's unlikely to be one particular nutrient, because you won't tend to see the same nutrient causing a problem on both acidic and alkaline soils.
Thank you, Barry. Question 2 that I have: why does YCS appear to affect?
Claire, can I just add to that? Sorry.
Yep.
Yeah, that's all right. I just want to pick up there on and to confirm what Barry said about all the nutrient trials that were done. That is, everything there is absolutely 100% correct. And silicon, yes, did show higher levels in sugarcane that's already a silicon accumulator, and Sophia in our chemistry lab in Dripilli did a lot of that work with thousands and thousands of samples. So one of the things that we did find in response to stress, Kaithwath and Dunn, who did most of our gene expression work, she found that there was an overwhelming correlation between YCS and abiotic stress. But I'm not saying that it's only abiotic stress. Biotic stress can still can still lead to oxidative stress in plants. So we see a high association with stress, with YCS in all the samples that we've taken. We see an increase in the heat stress proteins in response to internal heating of the leaf that comes through that closing of the stomata that is hand in hand with the reduction in photosynthesis, or turning down the photosynthetic genes to try and reduce the intake of carbon dioxide. So you get that internal heat go up, and we see stress associated with that. We also see, as sucrose begins to climb and we shut down those photosystems, that there is an increase in reactive oxygen species that cause a lot of oxidative stress, and the plant will do a whole lot of things to try and quench that by producing antioxidants. So it's absolutely 100% confirmed that, you know, YCS is associated with stress, but it's just not abiotic. It can be biotic. And so what is it having the most effect on? And in this question here about nutrient and water, I said before, when we were talking about why the yellowing occurred and why sucrose was high in the leaf, was because that it wasn't a problem with the leaf, it was a problem with the stalk. Now, one of the biggest things, or one of the most important things for growing a large internode is water. That's an absolute given. And you know this because right now, if you're trying to ripen your crop for harvest, you'll take the water away. And that'll produce small internodes, and that will increase the sugar content in those internodes. So those internodes, as I said in the previous webinar, will grow for a particular amount of degree days or heat days, around about 380 degree days, and then they're set in concrete. They'll grow a little bit fatter during that time. They do most of the growing in the first 150, but that's all driven by water. And they have to have good nutrition as well, as Barry said. So water is a key driver there in terms of growing a large sink, or more sink tissue, or a bigger sink capacity to be able to accept all of the sugar being produced by the leaf during that peak photosynthetic period. So if you have the ability to irrigate when the crop is looking quite dry, and rather than holding off, then I would urge you to do that to reduce the water stress on the crop during that peak growing season, because you'll grow a larger internode capacity to accept more of the sucrose from the leaf, and therefore you'll have less accumulation in that leaf, and it won't rise above the upper tolerable level. And once it goes past that level in the leaf, that's what tips it, and it goes from green to yellow. Thanks, Claire.
Thank you. Thanks, Gerard. Okay. So the question number 2, why does YCS appear to affect particular varieties differently? My observation is that Q238 and Q240 have been badly affected over the past 2 seasons, and in many cases, the adjacent varieties can show little or no symptoms, same soil and same farming practices. I think Gerard's going to be answering this question.
Yep, thanks for that question. It's an excellent question, and I get asked this a lot, and also about other varieties, and I know many of you out there would be saying KQ228 is one of those, and Q208 and Q200, and many of the commercial varieties that are grown in abundance throughout the industry, from the very north to the very south, and even into New South Wales. Look, it's like people. We all have different tolerances to different levels of stress. So certain varieties will perform better on a particular type of soil, as you know, and under particular conditions than others. And you know which ones do or perform better on your farm. And you tend to favour those when you're making your choices. In terms of them showing YCS symptoms, it all comes down to accumulation of sugar in the leaf. I said just before when I was talking about stress, and we know this level, it's a 200, what we call the 200 level. It's 200 nanomoles of sucrose per milligram of dry mass of tissue. there seems to be about approximately the upper level that the sugarcane leaf can handle. That's what we found through our research. Once sucrose surpasses that upper level, that's when it tips it to go yellow. Now, I say that in general. It would vary between the varieties, but we've literally tested thousands of samples across all the varieties grown across the state, and it seems to be around about that 200 mark. So there's not a lot of difference. We've run variety trials, and there's been a variety trial that's been in field in the Herbert for 3 years, and it has 9 commercial varieties in there, and most of the ones that I just mentioned before are in that trial. And we see some variation, yes, between the varieties, and we're about to harvest that field again in the next month or two to get more data, the results of that will come out at the end of the year. But to date, we haven't seen any real difference in that upper tolerance level as to how much sugar they can accumulate. So to answer this question, I know it's a little bit long-winded. To answer this question, I suppose if you had a variety that had a predisposition for producing a high amount of sucrose early, and it just couldn't get rid of that from the leaf quick enough to a growing capacity in the internode or stalk beneath it, then it may accumulate that sucrose a little faster than some other varieties. But I've absolutely no evidence to support that statement. So all I can say to answer this question is that we see no evidence of it in the trials, but I do acknowledge that you do see it out there. The other point I want to make is that some varieties tend to look worse than others because they hang on to their leaves rather than shed them as they yellow. And I know I've been tricked a little bit by this before, and I go up and have a look, and then it's not so much the case. But I do take your point that it does tend to at times look worse in some varieties than others, and it may come back to just a fine tuning between how much they can tolerate in the amount of sucrose that's in the leaf. Thanks, Claire.
Thanks, Gerard. We've also, one of the questions that's been added to the chat function, I'll include that with this next question. My question is, if the start interviews are reducing the size of the warehouse, why hasn't it shown up before now? Oh no, sorry, that's the next question. Sorry, everyone. Acknowledging that the impact varies across regions, what can be done to enable farmers to live with YCS and remain productive? Also, William included a question, what is the yield loss due to YCS? I think Gerard is answering this question as well. So acknowledging that the impact varies across regions, what can be done to enable farmers to live with YCS?
This is an excellent question. I would say, like, this is a very difficult question to answer as well, but it's like, say, we don't find a vaccine for COVID-19. If we have to learn to live with it, we'll do things to live with it. I think with farming, and you know it's a tough game, that all farming practice has to meet challenges that are thrown out there, and YCS has been around for about the last decade. So what can we do? We know that its symptoms appear when the crop is stressed. We also know that it's driven by growth rate. So what can we do to mitigate YCS? How can we live with this within our crop, and how can we reduce the impact of it? Well, we have to have a look at how we can keep growth rate up during that peak growing period between December and March in particular, when there's the highest amount of solar radiation and the highest heat for growth. So we have to try and keep the growth rate up. So we need to do everything that's physically possible to do that. So all I can suggest is to follow best practice. It's not about putting on more nutrients, as Barry said. It's just follow 6 easy steps, but to try to reduce the stress on your crop if at all possible. That way you can get the highest amount of growth when it's needed, and therefore it'll be able to, you won't end up with a source sink imbalance, with a supply-demand imbalance, which will cause the yellowing in the leaf. I think that's that question. Oh, there was the question there, Claire, about yield. Is that correct?
Yes, that's correct. So William asked, what is the yield loss due to YCS?
Okay. It varies, and in some of the trials we saw as much as 30% in the past, and in the same trial run more recently, there was absolutely no yield penalty. So why such a big variation? I'm crunching a lot of numbers across 7 years of research, and the outcome of that will be at the end of the year. But from a myriad of trials that have been run, but I think the answer to that question is all about, and I mentioned this in the webinar, is about not so much about YCS severity, it's more about YCS longevity. How long do you have YCS in your crop? I said before that YCS may start in a leaf 3 or 4, then it moves up the canopy. If it hangs around and it affects 5 of your leaves and you have a 50-leaf canopy over the course of your crop cycle, then you're going to lose 5 out of 50 leaves, which is 10%. But if those leaves only run at 50% capacity because half the time they're yellow, then you're going to lose 5%. So you could have a 5% reduction in yield. It's all proportional to the amount of time there is yellowing and how much yellowing there is. That's the only way I can answer that question without hard data. But we do see a high variation. But in the majority of cases, we only see yellowing for a 4, 5 leaf, 6 leaf at the most period. But we do see it in extreme cases around for many weeks. And that's when you're going to have your highest yield impact.
Thank you, Gerard. The next question is, my question is, if the short internodes are reducing the size of the warehouse, why hasn't it shown up before now? You couldn't have gotten any shorter internodes than Badilla.
Yep, that's true. They have very short internodes. The thing to remember is to not compare across varieties. That's important. Compare within the variety. So if you look at a variety like Q240 and you're seeing whether there's been any slow growth, or any variety for that matter, you look within the same variety and compare the internodes. They should all be approximately the same size if growth hasn't slowed down. So when growth slows down, you'll see the internode length shorten, and then when growth speeds up again, when there's more moisture or better climate, you'll see it elongate, and you'll be able to pick the periods along the stalk where it has slowed down. So small internode, then a large, small. And with YCS, that's when we see the expression occur, because we get a small internode from slow growth and then we get rapid growth. And it's that period there that you see it. So it comes after the slow growth, not before and not because of. So YCS doesn't slow the crop down. It's the crop slowing down first that then leads to the sugar buildup in the leaf that causes the expression of yellow. It's not the reverse of that. So in Badilla, yes, it has lots of little short internodes, but that's normal for Badilla. That's for its leaf, produces sugar, it can accommodate that because it's also a very thick stalk. So it's just not length that you want to look at. You need to look at the total volume of the internode, its total capacity to store. So it can be short and fat or long and thin and still have the same volume. But the important point is to compare internodes within that variety. And then if you see it shorten down, you'll know it's slow growth. But if they're all the same, then you know that the growth rate has been maintained. I think Rob might wanted to say something about that.
Yeah, thanks, Gerard. One of the things I'd like to emphasise here, which I think is important, is that yellow leaves are a bit like the safety shirts we wear, the high-vis equipment, and it takes our main attention. So yellow leaves are very easy to see. And one of the things that Gerard's been pointing to is the fact that when the stalk starts to slow, but the leaves are still photosynthesizing, then you get an accumulation of starch and sucrose, and it affects the leaf color and the leaves. But it's actually an interaction between other factors as well. So when you see yellow leaves, it's not necessarily just one condition that you're looking at. There may be other factors that are contributing to the slowing of the growth and the accumulation of sugar in the leaves. So when you see yellow leaves, be aware that other factors may be also affecting growth, which may be making the severity of YCS much higher than it normally would. So I think it's important to, not the yield loss associated with YCS may be associated with the yellow leaves, but it may be also an interaction with other conditions. As Jared's pointing out quite rightly, that Badilla has short internodes anyway. And so it's not the short internodes, it's the fact that the growth slows in varieties that normally have long internodes. That's the main issue to consider. But don't blame everything on YCS. We need to try and sort out what the yellow leaves are, how they're affecting growth, and are there other conditions that are interacting with YCS that are actually leading to poor yield as well. Thanks, Jared.
Thanks, Rob. Back to you, Claire.
Yeah, thank you. We have a question from Jordan, or a couple of questions from Jordan. So you might like to ask this question yourself, Jordan, if you go off mute. He's got a few questions. How do we manage if this YCS occurs on the farm? What are we going to do? How to prevent the YCS in the high rainfall areas and in the dry zones? And also, why didn't this occur 10 to 15 years ago? Because we have more varieties, we're changing our farming systems, or climate change. Why? So thank you, Jordan. Who on the panel would like to answer this question?
I'll start off. In the webinar, I did put up, a couple of hypotheses. Look, everyone's got their own theories as to why. I just simply presented one of those scenarios, which came from the bomb site, about what's happened in the last decade in terms of temperature and how it's changed since the mid-1960s. We've had about a 1.5 degree increase. Now, I probably didn't put that point across very well. Wasn't so much talking about just the 1.5 degrees being the thing that caused the stress. 1.5 degrees in terms of, while it doesn't sound like a lot, does cause a big change to climate. And in the last 10 years, and I had some graphs there in the webinar that showed when the severe weather events occurred and cyclones and whatnot, there's been some very, very extreme periods. And in fact, you've gone from very, very dry to very, very wet. And I know you probably say, yeah, look, we've had that before, but there's been an exceptional amount in this past 10 years. And I think if you look at the data as well, the Bureau is now saying that we have an extra 4 weeks of summer and 3 weeks less of winter than we did 10, 20 years ago. So things have changed. They mightn't stay that way. It might go back to what it was. I'm not sure. I don't have a crystal ball, but there's plenty of theories out there. But one thing is for sure, something has changed. Something has led to a decrease in growth during this period, and potentially there's been a little more sucrose even being produced from the leaves. That imbalance between the supply from the leaf and the storage in the stalk has been upset, and that's why we've seen the yellowing occur. So if we can identify what's caused the slowing down, then we'll have those answers. But I don't think it's a simple single answer, not a single cause, because if it was, we would have found it. And climate's very complex, and that's one that I put up there, but there's probably many others, and some people on the panel might want to throw in their views about that.
Yeah, Jared, I guess, sorry, it's Barry. Yeah, I guess I support the what you're saying about the extreme conditions, and I know for those who don't know me, I'm based in the central region. We have had a drop off, or it seems we've had a drop off in that sort of spring rainfall. So our springs have been really dry, and then often they're followed by quite heavy wet seasons thereafter. Now whether or not that's a trigger or not, but it is something that we've been experiencing quite a lot.
And I think to go with that, Barry, as you say, if you do get a very dry period, and I mean very dry, and you've got no access to irrigation, then you will produce lots of short internodes during that period. And then once you get good growth, everything's right. And you've got plenty of water supply, plenty of sunshine, you'll get maximum growth. And those leaves, they'll want to export, and they'll produce heaps of sucrose, and they'll want to export it. But there's just nowhere for it to go. And that's why we see the crop grow out of YCS. It takes a time, because it has to move up through those developing internodes before that balance comes back in sync between the supply from the leaf and the storage in the stalk. And that's why we see it grow out. But as Barry said, that's the typical cause of YCS, that dry period followed by a good wet period with plenty of sunshine, all the ideal conditions for producing sugar. Now, what can you do about that if you've got no access to irrigation? Probably very little, other than trying to drought-proof your property. And I'm not sure how you do that, but there are mechanisms, farming mechanisms that you're already probably using with trash retention and those sorts of things.
Thank you, Jared. And Barry, are there any more questions from guests at all? I think Sam has gone and put some links on the chat function, and I'll hand over to Brad.
Claire, I think Serge was put his hand up there.
Oh, I can't see that. Sorry, Serge. Serge, have you got a question? You're on mute at the moment, Serge.
People would say that was a good thing, Claire. Just on the yellowing and thing, there's a block here that I've had a problem with, and it's Claire's sent some photos through to you guys, and it's been commonly believed, I think, that it's chlorotic streak. Now, could you elaborate a bit more on chlorotic streak? And I can, after the information that Claire's giving me, I can actually just about trace it back to Cyclone Debbie. So I'll be interested in your thoughts. Thank you.
Rob, I go to you.
Yep. Okay, thanks, Jared. Yeah, Serge, I can talk a bit about chlorotic streak. So it's not surprising that it was associated with the cyclone. It's caused by a protozoan, which is a little tiny organism that lives in the soil, and it's spread by floodwater. So wherever there's either poor drainage or lots of rain or floodwater, then this disease will spread into the cane in susceptible varieties. It's one of the reasons why we get approved, because hot water treatment can eliminate it. It actually lives in the cane stalk and the roots, and so it's important to get cane that's been treated to eliminate it from the cane. Unlike RSD, it can enter the cane just from living in the soil. So when you plant it into an infested ground, it can actually go straight back into the cane. So it's difficult to manage from that perspective. So the main thing with chlorotic streak is keep getting disease-free cane. Ensure that you've got good drainage, the best possible drainage you can have, and then it's not transmitted by harvesters or cane knives, so it's not like RSD in that regard. So the main thing is disease-free cane, improve your drainage as much as possible, and then there'll be a few varieties which are a bit more susceptible, and your local adoption officer might better help you out with that. For instance, Q238, it gets more clotting streak than some other varieties. If you need more information, contact your adoption officers or the local productivity board staff. They know a lot about it. And if nothing else, you can get back to me as well. Does that answer your question, Serge?
Yeah, it does. I've been putting it down to yellow canopy. Last year I had a 100-ton condemned out of this one block, and it's been problematic for, as I said, probably since I can trace it back to 2017. It hasn't lasted that long, and yeah, so yeah, thanks for that.
Yep, no worries. No worries.
And thank you, Serge, also, because we did take a photo. That photo was just showing was Serge's block. Serge did get in touch with us after the YCS and was able to help Serge to identify that not everything in the cane is YCS. So are there any more questions? Okay, Bill, I think, has got a question. He's raising his hand there. Yes, Bill. I think you're on mute there, Bill. Bill, would you be able to type, because I can't hear you?
Yeah, sorry, Bill, we can't hear you because I think you've gone back on mute, but we couldn't hear you when you were off mute either.
We might have to come back to you, Bill. Sorry, after the meeting, we couldn't catch any of that.
We do apologise, Bill. Unless you're able to type your question, that would be great. Is there anyone else with their hand up or able to type a question for the panel? But thank you everyone for joining us today. And Brad, would you like to?
Yeah, thanks, Claire. No, I just want to thank everyone for coming. And if you do have further questions, by all means, you know where we are. Contact your local office or your local adoption person, and they can put you in touch, or access the SRA website. Barry was just going to say a very few concluding remarks, and we'll leave it there. Thanks, Barry.
Okay, yes, and yeah, just to support the others, and thanking you for participating today and for asking questions and providing questions, prior to the actual meeting. I guess we do realise that this issue and particularly YCS is a concern for people, and particularly for those that have got blocks that are badly affected by it. So we do realise that. And also that we don't have all the answers. We've obviously got, we understand the process quite very well in terms of why the crops are turning yellow and through the source sinking balance. But I guess the triggers for why that source sinking balance is probably an area that we don't quite understand fully. We obviously think that stress is associated with it, but there might also be other things that are triggering that source sinking balance. But once that imbalance is in place, then I think we've got a really good understanding of why those leaves turn yellow from there. I guess what we were pointing out today was that there are other reasons why crops can turn yellow, and I suppose what that really means is that early detection of what is actually happening in your crop is key to this, and it's certainly key for for diagnosing YCS. So the sooner you can identify it, and the sooner that you can bring it to the attention of trusted advisors, prod boards, SRA, etc., the more likely it is that we'll be able to identify it, and also, potentially important component is to track its progress from there as well. I know people have submitted photos to us where crops are looking in really bad condition, but one of the issues is that they may have multiple secondary things going on in those crops, and so it might have all started with YCS many months ago. Now when we're looking at the photo, it looks like there might be multiple things happening. So early diagnosis and tracking the progression through those crops, I think, is going to be really important. We didn't get to every question, so I apologise for that. Some people submitted a large number of questions, and we'll be going back to those people and following up with them, probably with some written answers as well. So once again, thanks for everyone participating. Hopefully this webinar and the previous YCS webinar, like I said, they don't have all the answers, but hopefully it's assisting in the process of trying to understand what's actually happening with these crops and eventually getting to an outcome. But thanks a lot.
Thanks, Barry. Thanks, everyone. This will be recorded and available on the website later in the week.